Wonderful. Well, thank you, everyone, for joining us today. For those of you that don't know me, I'm Marc Hurlbert, the CEO of the Melanoma Research Alliance, and thrilled that we're joined today by a guest, Dr. Boris Bastian of the University of California, San Francisco. Excited that today we'll have the next of our MRA meetups, an update on acral melanoma research. And really looking forward to hearing from Dr. Bastian and asking him a few questions and then opening the floor to any of you that might have questions. Just a couple of housekeeping things before we get started. We want everyone to know that this is a public webinar, so we are recording this for patients and families that couldn't join today. This will be recorded and shared on a private channel, but it is a public webinar, so we are recording this. If you are not speaking, we ask you to please mute yourself, and Renee and the team will help us just so we keep noise down to a minimum. And really excited to host this webinar today. And just one reminder, when you want to ask a question later on in the session, please raise your hand on Zoom, and then I'll call on people one at a time, and the team will help me unmute and mute people. And then lastly, this is an educational session. We really urge all patients and caregivers and family members not to ask for personal medical advice on today's webinar. So with that, why don't we let Dr. Boris Bastian introduce himself. He is an internationally renowned dermatopathologist, dermatologist, professor of dermatology and pathology at the University of California, San Francisco. But Dr. Bastian, why don't you say just a little bit more about your background? Thanks for joining us today.
Well, good morning, everyone. It's a pleasure to be here. Yeah, you said it. I'm a skin pathologist and trained dermatologist and mostly melanoma researcher here at UCSF in San Francisco. And over the last 20, or I'm afraid almost 30 years or so, I've spent time on studying different types of melanomas and what distinguishes them. And I'm going to share a little bit of that with you today. Can I share my screen, Mark?
Yeah, that would be great. Do you want to pull up your slides? And while he does that, just excited to have Dr. Bastian walk us through sort of how the various different types of melanoma are different, similar to or different from each other. So Dr. Bastian, the slides have pulled up and they look great.
Okay, so just a brief primer on cancer in general. So cancer is a disease of the genome, a genome of individual cells in our body as we live our lives and get exposed to environmental or intrinsic agents that mutate, change our DNA. Our cells accumulate mutations through our lifetime, and usually we are as long-lived organisms. We have very robust barriers to have not our cells behave in a rampant way, and usually for a cancer to form, one and the same cell has to accumulate multiple independent mutations to break through all those safeguards that keep our cells in check under normal circumstances. And so this here is imagine like a switchboard of a cell. This is supposed to be a pigment cell or actually a melanoma cell in which a variety of signaling pathways have been disrupted. So you may have heard about some of the genes associated with melanoma, such as BRAF or RAS or KIT or others. They operate in signaling pathways, and they get corrupted through mutations where they get slightly changed and don't function as they should, like a stuck doorbell or stuck gas pedal, if you want. And the red ones here are the ones that basically are increased in function by these mutations. So they are like the stuck gas pedal. And the blue ones are braking genes that oppose the action of the red ones, if you want. And those also get disrupted by mutations that kind of perturb their function. And so when we look at melanomas, and we often find mutations in any of these pathways. And then often one of those is sufficient to activate this pathway here. So you usually have either this one or that one or that one, because they all activate this pathway. Um, and these are these kind of mutation 1, 2, 3 that I was showing the first slide. As they accumulate, as the tumor evolves from something very indolent to something that can potentially be dangerous. And the processes that actually cause these mutations vary dramatically depending on which type of melanoma we're talking about. The melanomas that occur on the uh, skin that is exposed to sun are mainly caused by UV radiation from the sun. And then there are kind of just random mutations that occur as cells divide, as we age. Cells have an active metabolism. They generate reactive oxygen and things like that that can just randomly change the DNA and cause mutations. Then there are so-called kind of clock-like mutations that just accumulate with time over our lifespan. And then there's some emerging evidence, and I will make some reference to this later, that physical force, so actually squeezing or traumatizing cells, could also cause the damage in the DNA. And so just to illuminate this, or with a few examples here, if you just look at a simple sentence, these point mutations, they're kind of like typographical errors where individual letters in the DNA get changed. So this would be a point mutation here, and those are the type of mutations that are typical for UV radiation. There's then other types of mutations where you get actually pieces of DNA get repeated more than they were in the original or less. And so here would be a duplication, and you will see, especially in melanomas like acral and mucosal melanomas, we see sometimes repetitions of more than 100-fold of certain passages in the genome. And then there's something that we call structural rearrangements, where things here get repeated but also turned around. So you see here jumps over in reverse. So this is an inversion. And then here you can also see that sometimes the meaning gets completely jumbled because pieces of information get rearranged. So these are qualitatively different types of mutations, point mutations, versus these copy number and structural rearrangements. And I show you this here, kind of a depiction of a real genome from acral melanoma, how we show it. And you can see the entire human genome is shown with all its chromosomes from 1 to 22 and X and Y in this circle. And these little red, line shows whether the copy number of the segments is normal or increased. Here where you can see this higher level. And just for reference, the human genome is very large. This, to go all the way around here, takes 3.2 billion letters. And each of our cells have 2 copies of this. So we have like 6.4 billion letters of DNA letters equivalent to these letters here in each of our cells. So that's almost as many as we have humans on the planet right now. And what is happening in this genome is you can see that there are pieces of DNA here from chromosome 5 linked to another chromosome, and these are these gray lines, and then there are orange lines that link pieces of chromosome 5 to chromosome 5 itself. So this is how this really looks. You see chromosome 5 here, and you can see the copy number should be an even line, and then we have these little bits of DNA that are highly repeated here. So this accounts to probably 60 or so copies of this. So these are these duplications, but then we have these many um, rearrangements where things are jumbled. Like here, the orange lines indicate jumbling between two different positions in the same chromosome, and the gray line between these two different chromosomes. So this is basically just a close-up of this phenomenon here. So you can imagine this is like a severe alteration of the genome, but interestingly, it occurs only at a few sites in the genome, and this is very characteristic of acral and of mucosal melanoma. So when we compare these two types of acral and mucosal melanoma, I've depicted this here, making the point that their epidemiology is very different. Acral and mucosal melanoma basically affects all world populations relatively equally, irrespective of skin color. Whereas the melanoma that is caused by UV radiation is primarily a disease of people with light skin, poor tanning ability that have less effective shielding against UV radiation and therefore accumulate these point mutations, these single letter mutations at a higher rate. And so that's shown here, kind of the typical genomic damage that you find is just hundreds of thousands of typographical errors, point mutations in the genome. And there's like two major types of melanoma of this type. One affects the heavily sun-exposed sites like the head and neck, the outer arms, where one gets a lot of sun over the lifetime. And these melanomas increase with age, and they have a characteristic pattern of mutations that are the equivalent to the stuck gas pedal I was introducing earlier. And there's a second type that primarily affects individual patients under 50, and that arises on skin that is not so heavily exposed. And they have a lower mutation burden. These here often arise from pre-existing moles. So these moles that arise during childhood, early adolescence, later acquire more mutation from the sun, and the mole turns into a melanoma. This is very different. In these melanomas, we do not see the typical point mutations associated with UV exposure, and instead we see these characteristic changes, these breaks in the DNA where two pieces are put together in the wrong order that I showed you in the prior slides. And so no scars, so to speak, in the DNA from UV exposure. And they also tend to have a different pattern of mutations that kind of are the main oncogenes, we call them, that the gas pedal equivalent in that drive these cells. And those also tend to arise or become more frequent as we get older. And these happen to be the most common type of melanomas, both of those, in patients of color, but not because they're more susceptible, but actually just because in people with dark skin, as I said before, you have much better shielding against UV radiation. So they actually very rarely get these melanomas, so that these are relatively more common but they're absolute numbers. If you look at the number of patients per 100,000 with acral melanoma, they're very similar throughout all world populations. So just to recap this real quick, so this is on the sun-exposed skin. We have these two types. One is that arises on these chronically exposed sites. They're now called high cumulative sun-induced damage melanomas, high CSD in the WHO classification. They often arise from a flat brown spot that we call melanoma in situ. And the order in which mutations arise, as I showed in the first slide, mutation 1, mutation 2, mutation 3 is like this. And so you have this gas pedal mutation first, and then the brakes get corrupted, and then you become a melanoma. The second most common type that I mentioned on light skin is the one that arises from moles. So you actually, or this is the mole, and this is the melanoma that grows out of this. This is the one that has a slightly left-shifted age distribution. The main difference to the one before is that it seems like here the order in which breaks get broken is slightly different from the one before. And then we get to the ones on acral skin. So acral skin is the palms, the soles, and the nail apparatus, or the mucosa, which can be the mucosa of the head and neck, so mouth, nasal cavity, the genital, perianal areas where these melanomas can arise, sites that get very little or no sun exposure at all. And here, at least in this one, we studied this recently. There is a peculiar inversion of in which things get altered, with this alteration happening first, where it happens later in the others, and the actual kind of gas pedal or oncogene mutations happen later, which creates complications for treating those as targets, because often you find tumors from a single patient where one part of the tumor is driven by one oncogene and the other by a completely different oncogene, where you basically would have to target both to really effectively suppress the patient's melanoma. And this is another example here from a patient we saw a while ago who had seemingly two separate melanomas on her palm. And so this would be a typical acral melanoma. And by genetic analysis of this kind of pretty thick melanoma here, we found this, what we now call hailstorms, this focus in the genome where everything is reshuffled and you get this extra copy number here. And we actually so this would be the site of the jumps here in the sentence where it gets repeated many, many times. So we actually made kind of a little fluorescent probe against this and just try to count the copies of this in each of the patient melanoma cells. And you can see the red color here is equivalent to one of those probes. And this is a cell from here. You can see it has a lot of copies from this locus. But this melanoma here that seems to be completely unconnected had the same, or actually lower number of copies, but the same alteration, this elevated copy number at this site. But the point why I'm showing this is that also the normal skin in between here had cells that also had this amplification. In fact, we could map this out. This is the white line here, that these two melanomas were not actually two separate lesions, but they arose from a field of cells that already carried this alteration and then kind of both evolved at different speeds, like an iceberg that is submerged underwater. And you see just two tips, but in fact, they're actually connected under the surface. And so our work over the years has revealed these alterations, these hailstorms, these characteristic reshufflings at certain parts of the genome as a very early feature in acral and probably also in mucosal melanoma. We're just investigating this, so we're not entirely certain about this. But in acral melanoma, we have very clear evidence from examples like this, that this is a very early change that initiates these melanomas. And the question is, what causes these hailstorms? One thing that others, several studies from Korea and Japan have noticed, showing the distribution of acral melanomas on the sole, and the redder it gets, the higher was their count of melanomas, that this coincides with the weight-bearing areas of the foot. So this is a pressure map from the foot, suggesting that maybe physical force impact could lead to DNA damage. And there is some precedent for this from work. We can get this to work. This is a study where we're just looking at DNA here colored in green in the nucleus of cells that move and basically get squeezed through a narrow space. You can see there's this little green blob that is popping out here. This is what we call a micronucleus. And we know that these are actually sites where this chopping up of the DNA and reshuffling of the pieces can happen. So there is actually experimental evidence that physical force can lead to DNA damage. I mean, I have to really emphasize at this point, this is circumstantial evidence, but there is some epidemiological evidence that the pressure-bearing sites have more acral melanoma, and then these types of DNA damage that I showed you would also fit with what we expect to occur when the actual mutational mechanism that is causing the genomic damage is physical force. So this is kind of in a nutshell the difference between these melanomas on sun-shielded sites and sun-exposed sites. We believe that they're really arising from two different types of pigment cells. There is now some biological evidence showing that the pigment cells in these body sites differ from those elsewhere, and they may have just different states, different vulnerabilities, probably explaining why they have a slightly different pattern of mutations in their switchboard that I showed you earlier. And I think that was my last slide here. I now invite you to ask questions to clarify some of the things that I tried to express. I hope I made it reasonably clear so that you could follow along, but please don't be shy and feel free to ask questions.
Yeah, no, thank you, Dr. Bastian. Really a wonderful presentation. A few questions already have come in. Um, Wanted to just first of all give you a huge kudos. I know that you won't appreciate me putting the spotlight on you, but your team and your lab at UCSF is one of the few around the world that have really defined the genomics of acral and mucosal. Maybe 5 or 6 labs across the entire world that really study this deeply. And for those on the call that don't know, the U.S. federal government about a decade ago published the Cancer Genome Atlas on cutaneous melanoma back in 2015. And really, Dr. Bastian, you and your team's work has really helped us decipher acral and mucosal. I wanted to, and I loved your analogy of the two types of mutations, one that gets the gas pedal stuck on, and then one that the brakes start to fail, and you need a sort of a combination of both. One question I wanted to ask was for cutaneous melanoma, the low CSD version, even though that's lower chronic sun damage, these patients, it is still caused by UV damage, right? So it's still a UV irradiation causing that?
Yeah, so definitely there's very strong evidence that the changes that turn the mole into a melanoma, I mentioned that most of those arise from moles, right? That those mutations have very strong features of UV radiation. I showed the typographical errors. We can tell from the type of typographical errors, especially when you see many of them in a cancer, as we do in those, we see 100,000 or so. What is the likely mechanism? So, for example, tobacco smoke would have a very different type of change than UV radiation. So that's how we can tell. And yes, those clearly implicate UV radiations very strongly, at the very least from the transition from the mole to the melanoma phase. but probably also the moles themselves are initiated by UV radiation.
Great, great. Well, wanted to ask one question that came in, just asked if whether acral patients with acral or mucosal do respond to these newer immunotherapies, checkpoint inhibitors, and if there's any data around sort of their response.
Yeah, they do respond, but in general, not so well as the ones from the sun-exposed skin. Part of this has to do with what the physicians call mutation burden, meaning how many mutations are actually in the cell. And you have to imagine that most of these mutations actually do nothing. It's like scratches on the paint of the car or something rather than disrupting the brake system or um, but they alter some of them, a subset, even if they don't make the car behave in a different way, they alter the way how the immune system sees it. And so the more of those mutations you have, the more likely it is that the tumor actually had an active interaction with the immune system and somehow found a way to evade the immune system. And then these immunotherapies basically um, make the immune system active and able again to kill the tumor cells. So that's when you have a high mutation burden, higher visibility to the immune system, better response to immune checkpoint blockade. This is how these are called. Acral melanoma, as you could tell, has, first of all, much fewer point mutations, sometimes very few, but it has these other things. And this hasn't been so well studied, in part of their very complicated nature that the current DNA sequencing methods have actually not been able to fully decipher what's happening there. We're just starting with that, but probably creating much fewer targets for the immune system to work with. And so um, there is response, but usually not as pronounced as with the others.
Thank you. Chris, you want to ask your question live? And anyone else that wants to ask a question, you can type it in the chat or raise your hand. Chris, the floor is yours.
Hey, kind of piggybacking off of that, with the difficulty in managing all the information of the genomic sequencing, are either you or your colleagues studying or implementing AI or kind of machine learning and analyzing some of the genome copy number or sequence changes, or using it for predictive diagnostic or treatment elements? And if not, are there, where could I learn about that? Who's studying that or how that's being implemented?
Yeah, that's a great question. I would say not yet. We're using a lot of AI just to help us condense information and so on, but really to understand this information. Um, there aren't any really good models yet. I mean, obviously, AI is based on these large language models, right? And we don't really have a large quote unquote language model for the DNA yet. But this is a very hot topic of investigation where you basically, because it's also a form of language, if you want that, like in my analogy there, to understand the genomes better and make predictions that with our as humans or would be very difficult for us to do. That is mainly happening kind of on normal genomes from organisms to kind of look at a piece of DNA and say, oh, this animal must have had antlers or something, because you can read it in a way that would imply that. To analyze cancer genomes in that fashion would be really complicated because they are kind of one-offs. They're one-off perturbations of the human genome, and but I'm convinced that at the velocity this field is emerging, that we will be using this quite soon.
Yeah.
Yeah,
Thank
yeah,
you.
great question.
Great question. I wanted to ask, in your research, in clinical practice today, patients with acral and mucosal, if they have a BRAF mutation, could get BRAF or MEK inhibitors. almost all of them get immunotherapy if they have stage 4 or advanced stage 3 disease. But on the research setting, in your research on these structural rearrangements and copy number variants, are there any interesting, what you consider novel targets that might be worthwhile for drug development or exploration?
Yeah, thanks. There's a lot to unpack there in this question. I mean, the BRAF, one of the findings I briefly went over, that these mutations like in BRAF or KIT that are therapeutic targets in acral melanoma arise later rather than as the first thing in the other melanomas, brings a complication for their suitability as targets. We were very surprised to find that we find melanomas where one half of the melanoma is driven by BRAF and then the other is driven by KIT, and they don't overlap. And this is something that we never see on these sun-exposed melanomas. And so that just makes it more difficult to use targeted therapies in those. The main question was, can we exploit some of these mechanisms that cause these hailstorms, this shattering of parts of the genome? That's why we carried out this study, seeing whether this is kind of an ongoing thing that is preserved as the cancer evolves, meaning that more of these shattered parts of the genome arise as the tumor goes from early stage in the skin and then starts spreading to other sites. And the answer was no. It seems to be it's a hit and run type of thing where this assault on the genome that is causing these alterations is really operative only once and probably kicking off the whole thing. So we were hoping to find some kind of defect in the cells that would make them vulnerable to some therapeutic interaction. Like, for example, in breast cancers in patients with BRCA mutations, they have a compromised DNA repair pathway that is causing mutations by itself. And that makes them, the cancer cells specifically, not the patient, fortunately, susceptible to these PARP inhibitors. So basically the cancer cells have an inability to repair certain DNA damage, and they use a fallback mechanism. And if you inhibit that fallback mechanism, then the cells die. We're looking for something like this in acral and mucosal melanoma. I think we have now a much better understanding of what the type of damage is, but we're looking at the aftermath, or have to kind of, like an archaeologist, work backwards from the alterations to see what actually caused it, right? But we do have a lead that we are pursuing that points towards a unique dependency of acral and mucosal melanoma cells that hopefully can be developed into some form of therapy.
That's great. That's exciting. Exciting. It's still at the research stages.
Absolutely.
Another great question has come in. Does the location of the acral melanoma have any influence on the mutations you see? So if it's on the palm of the hand versus the sole of the foot or underneath the fingernail bed, are there differences in location?
We haven't found this. We've looked at hundreds of cases that were published by other groups and analyzed in our own lab to look for patterns, also of different ethnicity. If you look patients from Japan versus Africa or South America, we did not see striking differences there. So it all points to this is a pretty homogeneous disease with mucosal melanoma. There may be some differences between the head and neck and the anogenital melanomas, but it seems to be subtle. The key changes of these kind of hailstorm shattering of the chromosomes are the same through all of them. It's a little bit of variation of which mutations are more common, but it's not a strong pattern. So we think those factors have a relatively minor effect.
Great. Well, great question. So we have time for maybe one or two more questions. If anyone else wants to ask something, either raise your hand or you can type it in the chat. But this has really been informational and educational, even for me, and I know most of your projects, and I appreciate, Dr. Bastian, you summarized a decade of your lab's research and probably, I don't know, 10 or 15 scientific publications in about 6 slides. So thank you for that, digesting it in a way that all of us could understand. Any other questions from the audience?
If I could jump in real quick, we gave people the opportunity to ask questions when they were registering. And this was actually something that you touched on earlier on in your slide deck, where you mentioned trauma as precluding acral melanoma sometimes. Is there a distinction between like repeated trauma in, say, somebody like a runner versus one big trauma event?
Yeah, you know, that those are the same questions that come to my mind also. I mean, the caveat with this is we don't have really strong epidemiological evidence that, runners are more susceptible to this. And so, I mean, if physical force is really causing this, it must be a very, very inefficient way. And that there's just kind of doing this all the time, and most of meaning this being breaking the DNA of some cells, but then those cells would die, and would never notice this. But every once in a blue moon, something happens that shatters, jumbles the sentence just in a right way that cell hangs around. Whether then it requires a second trauma to it could be, but it's totally hypothetical. That's where the archaeology event or analogy is actually, I think, pretty good, because you don't know whether these two ruins or that you see in the genome, which were caused by an earthquake, one earthquake or two different earthquakes. We don't have that resolution yet, but the fact that they are kind of have their parts connected to each other indicates it was one earthquake, right? Not two earthquakes. But these are things that we need to untangle.
Great.
And
There's
I
a
see
few more questions in the chat.
Mhm.
So
Yeah,
you
I
want to try
just
to
saw that.
answer those, Boris, about the pattern of ALM, acral lentiginous?
Yeah, so does the growth pattern of acral make it more prone to recurrence? I mean, recurrence is used in several ways in the field. The recurrence where it probably does matter, I want to talk about, is the one that relates to the primary melanoma growing back after it seemingly has been completely removed. You remember I showed the palm of the patient's hand, and there the outer line was actually the surgical excision, and you could see that field that we found with this method extended focally to the edge.
Oh wait, I already checked that. So
Somebody is not muted there. So you can imagine that this looks like completely removed by pathology review, but there is this subtle occult field that goes to one edge. So it's possible that that could spawn a recurrence later. We think that those cells are the very earliest manifestations, so it may take decades for this to actually become clinically manifest. And so we're not so worried about this type of recurrence. The other recurrence would be the melanoma actually goes to the lymph node. I mean, that's more a progression than a real recurrence. And so for the first part, I would say yes, the field effect and this kind of subtle nature of the early melanoma makes it more likely to cause a local recurrence, but not a distant recurrence. So I hope I answered this one. what gives you the most hope for acral melanoma research and treatment? Well, I mean, it's really that we begin to understand better now to understand the genomic alterations. And there's some major breakthroughs in DNA sequencing. Well, my analogy is that of a puzzle. You know, I told you how big the human genome is, 3.2 billion letters, and so far we could only sequence 200 letters at a time. And so if you then want to put the genome all back together, you would have tens or hundreds of millions of these little pieces that you would have to put together. And then there's lots of the genome is like blue sky, where the pattern looks the same. And so it makes it kind of very difficult to solve. You know, even with AI or computational methods that we actually do use for this, we can't fully solve the blue sky piece if all the sentences look alike. And that is important to put an acral melanoma genome fully together to see how all the pieces are connected to then infer what actually broke it. And now there's new technologies where we can sequence pieces of up to 100,000 base pairs. And so really large puzzle pieces, like in a kid's puzzle, you can actually, those are much easier to put together. And so we've just started to sequence acral and mucosal melanoma genomes with this new technology, and we're getting new insights how this all hangs together. And hopefully that will reveal some of the secrets of what is actually in there in these altered regions that make the melanoma go. And so that makes me hopeful for because the progress in that area is really picking up. So I think it will not take another decade or two before we make progress.
If you're up for it, Dr. Bastian, a couple of more questions. One is, what are your thoughts on some driver mutations, such as BRAF, appearing to be more common in females with acral melanoma than males? Any thoughts about gender differences?
Well, I don't think we have noticed this pattern. I mean, the thing about BRAF, especially BRAF, it gets very subtle. There's one specific mutation that is the most common, that is BRAF V600E. And that is the mutation that is most common in moles, and it's the most common mutation in these, what I showed as low CST melanomas. And we do think that some melanomas on acral skin are actually just these melanomas. They just happen to occur on acral skin, but they're not really acral melanomas in the sense that they don't have these hailstorms, these shattered parts of chromosomes. So, and one does get moles on acral skin. And interestingly, those moles have a very different distribution. They're kind of not on the weight-bearing areas, but they're on the arch of the foot. And so we think that those are apples and oranges. And so if we're talking really about BRAF V600E mutations, I would think that quite a few of those in acral melanoma can actually be attributed to melanomas of another type presenting on acral skin.
That's great. And then another question is amelanotic acral tumors different than melanotic acrals?
No, biologically, we don't think so. Obviously, that always creates a significant delay in diagnosis. I mean, if nobody thinks of melanoma, if somebody's nail is coming up and there's something growing and it's not pigmented, and people think it's a wart or something else, and that's the main problem with amelanotic melanoma. But biologically, we don't have any evidence indicating a significant difference.
And then I think the last question that came in directly to me was, there are some patients with mucosal and with acral that had a complete response either from just surgery or immunotherapy a decade ago, and they are what you considered like a long-term survivor after their initial treatment. And any thoughts about why some patients with acral or mucosal achieve long-term survivorship and others don't? Any thoughts about differences in responses to surgery and treatment?
Yeah, I mean, to surgery, it's I think obvious if you get the whole thing out before it could spread elsewhere, that's the best case scenario. I mean, we do get, when we do these comparisons of the primary to the metastasis, we get the impression that the spread happens very early. But still, you can completely get it out by surgery. The complete responses to immunotherapy are interesting, and the model there is that if you have a neoantigen, we call this, so something that in the tumor, in the tumor only, that the immune system recognizes as foreign. If that is something that arose very early as the tumor evolved, it would be expected to be shared among all the cells in the tumor because they just pass it on and on. And so if your immune therapy actually unleashes a response against such a neoantigen that is present in all the cells, then it could really completely wipe them all out. Whereas when you get neoantigens that get formed later, they're present in some metastases in the body, but not in others. You get partial responses. Some go away and the others continue to grow. That's a partial victory, but it's not enough. So you have to try another way to get at the remainders. But that would be my favorite explanation of how this could be explained.
Thank you. Thank you. Well, thank you, Dr. Bastian. I think I will bring us to a close. I'll just say for everyone that doesn't know yet about the MRA's Rare Melanoma Registry, if you're a patient with acral, mucosal, or cutaneous, I encourage you to join the Rare Melanoma Registry and share your data and answer some surveys and questions so that we can do more research like Dr. Bastian's work. The Rare Registry, Dr. Bastian, if you haven't heard a recent update, we have about 350 patients enrolled, about 150 with mucosal, 100 and some with acral, and about 100 with cutaneous. So growing an army of volunteers willing to contribute data and information. Thank you for all of your work on these rare melanomas, Dr. Bastian, and thank you for your time today. This has been really fantastic.
Yeah, and thank you all for participating and also for participating in rare melanoma. And so it really takes a village to solve this problem. Everybody coming together, sharing information and sifting through it. So I'm glad that we had an opportunity to share with you what participating in this way actually means for the researchers.
Wonderful. Thank you very much. Thanks, everybody, and have a wonderful day.
Thank you.